Okay, so now what we're going to do is we're going to start talking about pathotypes, all right? And I think we've already, it's a little difficult putting a lecture series together like this to present the concepts sequentially without discussing them before they're actually presented in detail. So we've already talked a little bit about pathotypes in the context of some of the presentations yesterday and this morning. I think you've heard the term before. This is what I showed you before. The lecture the lecture I just gave you, we talked about this, the forma specialis that were first described in 1896. What we're now going to do is talk about pathotypes, physiological races, that were first shown in a rust pathogen by E.C. Stakeman in the US in 1914. Okay? So this is the between-host specialisation, the forma species. What we're now talking about is within-host specialisation. So we're talking about one forma species, for example, Puccinia graminis forma species tritici, and the variation we see within that with respect to its ability to infect different wheat genotypes or different wheat cultivars. All right, so pathotypes, races, strains. These terms are all pretty much mean the same thing. So if you hear somebody talking about race analysis, it's pretty much the same as saying pathotype analysis. I tend to use the word pathotype. But strain, race, pathotype, it pretty much means the same thing. Now you can take different isolates of the pathogen, of a rust pathogen, like Puccinia graminis, forma species tritici, different isolates, and they may look different when they're compared for disease reaction on a range of genotypes of a plant species. So in other words, a range of genotypes of wheat in the case of Puccinia graminis tritici. These isolates that differ in their ability to, in their disease reaction or their ability to infect different genotypes are called pathotypes. And as I said, we can call them strains, we can call them races, we can call them biotypes. It all pretty much means the same thing. If the pathotype can cause disease on a particular plant, it is referred to as being virulent. If the pathotype cannot cause disease on a particular plant, we refer to it as being avirulent. All right? So you'll hear the term, and we'll recap this tomorrow, I think, recap on this tomorrow, but it's very important to think of those terms. If an isolate can attack or can induce susceptibility on a plant genotype, we say it's virulent for that genotype. If it can't, we say it is avirulent for that plant genotype. So, and as I said before, I've been doing this kind of work for a long time. Zach's been doing this kind of work for a long time. Dr. Prasher has a lot of experience with this. Dr. Javed has, and Sajid. There are a number of people in this room who have quite a fair bit of experience in identifying pathotypes. But what I'm often asked by people who haven't worked with pathotype identification is, how do you do it? And I say, well, if you look at a field like this, you can see that there's a yellow patch in the middle here. This is stripe rust. You can't determine what pathotype or race is present just by looking at a wheat field and seeing it's infected, right? You can't do that. They normally say to me, but you must be able to do it with DNA technology, you know? It's really, really high-powered, high-tech stuff we're told that we have these days. Surely you can do it with a DNA test. Well, you can't. You can't identify a pathotype with 100% accuracy based on a DNA test. Right? Can you do it with trap plots? Do people here know what trap plots are? Have you heard of trap plots or seen them in the field? Nobody's putting their hand. Anybody heard of trap plots? Yeah? Okay, so you put a series of genotypes in the field. Each one carries a different resistance gene. Example here, these are what we call the avocet nils. They were produced in my laboratory by my colleague Dr. Colin Wellings. You can put these in the field and some of them may be susceptible and some of them may be resistant. And you may know what genes they are. This is probably the YR1 nil, and I think this is the YR6 nil. So what you're looking at there is virulence on YR6, avirulence on YR1, but it's not telling you what pathotype is present. So you cannot tell what pathotype you've got by looking at trap plots in the field. They're very useful and very handy in scoring material in a paddock and knowing what virulences are present, but they won't tell you whether you have 1 or 2 or 3 pathotypes, and they won't tell you what virulence combinations you have in those pathotypes. What we have to do to identify pathotypes still, and we've been doing this for a long, long time, is to take a sample of rust from the field and put it on a differential set and then score what we call the phenotype of the rust. So if we're working on the rust, we want to know whether it is avirulent for a particular resistance gene, as shown here. So this is one pathotype that is actually avirulent. This is for leaf rust. It's actually avirulent for a gene called LR20. You can see a very small pustule there and the necrosis around that. And this is another pathotype on the same gene, LR20. It's virulent. But we do this, of course, not just with one resistance gene. We do this with many resistance genes, and that allows us to work out what the virulence and avirulence of a particular isolate is. And on the basis of that, we then give a pathotype designation. All right, so the DNA revolution is here, genome sequencing, microsatellites, all this kind of stuff. We're still stuck with doing seedling assays for pathotype analysis, and we're going to be doing it for quite a few years to come before DNA technologies supplant that, right? My experience, I've already told you, I've been doing race analysis for wheat stem rust, Puccinia graminis forma species triticide, for the wheat leaf rust pathotype, for the barley leaf rust pathogen, for miscellaneous grass rust, and actually since 1996 I've been doing this also on oats for the crown rust pathogen Puccinia coronata. So I've been doing this for quite a while, and I do that for all of Australia. And as I said before, there are a number of people in this room also who have significant experience in doing race analysis or pathotype analysis on rusts.